This is the basics of cellular organization. All animal cells are lined with a plasma membrane, which is a selective barrier that allows sufficient passage of nutrients and molecules in and out to sustain life. And these plasma membranes are made out of what we call a phospholipid bilayer. If you take a look at one of those phospholipids, you notice at the head, the little gray circle or elliptical shape is hydrophilic, and the long tail, two tails, are hydrophobic. And so they orient in such a way that the hydrophobic region, or the tails, point towards each other and are away from the watery region, from the aqueous region, which is outside the cell as well as inside the cell. And so they literally orient in the way that they are, not because of any kind of special attachments or bonds. It's just because, and so the phospholipid bilayer is fairly movable and so on. It's important to note that cells are tiny for a good reason. It's advantageous to be small. And the reason why is because as anything gets larger, it takes longer and more energy to get nutrients or products in or out of the cell from the center to the surface of the cell, or from the surface of the cell to the center of the nucleus. So it's an advantage to be small for that reason, for that transport reason. All of these organelles we will have to know. I will expect you to be able to recognize on a diagram. I'll be able to expect you to know the names and the functions and the relative location of each of these organelles. You got the nucleus, and the nucleus is wrapped around with a nuclear envelope, and that is fairly more or less in the center of many cells. And they have nuclear pores. And in the center of the nucleus is the nucleolus. Just wrapped around the outside of the nucleus, you have the endoplasmic reticulum, rough ER and smooth ER. Then just outside of that, you have the Golgi Apparatus. And then you have the rest of the organelles like mitochondria. And notice the difference between the animal cell and the plant cell. The plant cells are have a plant cell wall, and because they need to be they need to have pressure on all sides of the cell wall, they have a central vacuole that allows them to fill up with pressure, and so they're much more rigid than your typical animal cell. So starting with the nucleus, generally the most central part of the cell contains DNA, and and the nucleus is surrounded with a nuclear envelope, and the nuclear envelope is made out of nuclear membrane and the nuclear pores. So you have the nucleus in the center. Wrapped around is the nuclear envelope, and it's made out of the the nuclear membrane and nuclear pores. The nuclear pores is how you get things in and out, like RNA coming out, or certain proteins coming out, or certain proteins coming in, and then you have the membrane. Altogether, it's called a nuclear envelope. In the nucleus, you have DNA, and it's arranged in either chromatin or chromosomes, depending on what the cell is doing at the particular time. What we see in this example here is a bunch in the purple area, not in the dark purple area, but just around the outer cortex of the nucleus. You don't really see condensed structures. So chromatin, so what we see is chromatin, very relaxed, unwound DNA. That's what chromatin is. When the cell begins to the process of dividing, multiplying, the chromatin then condenses into what we call chromosomes. And so their chromosomes and chromatin are made out of the same stuff. It's just what form are they in. Chromatin are unwound, very relaxed DNA, and chromosomes are discrete, condensed, very obvious, small units. The nucleolus is inside of the very central nucleus. The nucleus is in the center of generally in the center of the cell, while the nucleolus is inside of the nucleus. The function of the nucleolus is that it makes ribosomes. The nucleolus makes ribosomes. And what is the function of ribosomes? Ribosomes help make protein. So the nucleolus makes the ribosomes, and the ribosomes are complexes that help carry out protein synthesis. And here we see an example. You see the two brown units, the large and small subunits of a ribosome. In the last unit of the course, we're going to see how this all comes together and exactly how does a ribosome help carry out protein synthesis. There is a number of organelles inside the cell that belong to the endomembrane system. The reason why they all belong to the endomembrane system is because they're made out of basically the same kind of stuff. Well, we already talked about the plasma membrane, and we know what the plasma membrane is made out of, or what most of it is made out of. It's mostly made out of phospholipids, arranged in two layers of phospholipids, and so we call it the plasma membrane. We call it a phospholipid bilayer. Well, it turns out that the rest of these parts, the nuclear envelope, the endoplasmic reticulum, the Golgi Apparatus, or the Golgi Complex, lysosomes, and vacuoles are all made out of the same stuff, phospholipids. And that actually helps the movement of molecules in or out of the cell, or into the nucleus or out of the nucleus, because these membranes are made out of the same things. They can bind and be released from each other from all these different endomembrane parts, because they can fuse together, or they can pinch off and leave as little envelopes called vesicles. So it's important to remember that the nuclear envelope, the ER, the Golgi apparatus, lysosomes, vacuoles, and plasma membrane, they are all part of the endomembrane system, which means they all make up they're all made up of the same stuff, phospholipids. Okay. Generally, when we talk about the different kinds of organelles, we start in the very center of the cell and we slowly work our way out, because that's how protein synthesis also is made. Protein synthesis, we begin at the center of the cell and we slowly work our way out. So if the nucleus is in the center of the cell, the next organelle just outside that is the endoplasmic reticulum, or the ER. The rough ER, generally we see first, and then the smooth ER we see second. Well, the rough ER is named rough because it looks like it's been pinned with these little bumps. These bumps are ribosomes that are attached to the ER. So it looks rough. It looks rough, so that's where it gets its name, rough ER. And the smooth ER is called smooth because it looks more smooth than rough. The smooth ER does not contain ribosomes. Okay, well, what's the function of these two things? Well, the smooth ER makes lipids, metabolizes carbs, detoxifies drugs and poisons, and stores calcium. Whereas the rough ER, which has those bound ribosomes, they secrete proteins that are bonded to carbs, or glycoproteins. They help make proteins, and they help distribute the proteins. They help transport the proteins along. How will they do that? Well, if you take a look at the ER in general, it looks like a little maze, and that is because the proteins, or the molecules that are turning into proteins, actually flow through that little maze, and that's how, with those ribosomes that help make proteins, the molecules travel through that little maze, and as it flows through, the molecule is being synthesized and getting larger and larger and larger, moving from the center of the nucleus through the rough ER, and it might go to the smooth ER as well. And again, the smooth ER synthesizes lipids and so on. So depending on the molecule, it might go from the rough to the smooth, or it might skip the smooth. And then what happens to that molecule? The next place it goes to is the Golgi Complex, or the Golgi Apparatus. So the Golgi Apparatus is just outside of the ER now. So if we take a look at the original this original slide that we saw all together, you take a look at the nucleus, the purple in the center, and then you have the blue ER, which is the rough and the smooth, and then just outside of that you see the Golgi Apparatus, which is sort of that teal color. So now whatever is made by the rough ER and by the smooth ER is sent to the Golgi complex. And so it consists of flattened membranous sacs called cisternae, and the functions is to modify the products of the endoplasmic reticulum, where the products just came from, which makes sense. It's going to modify now what the products that it just received from the endoplasmic reticulum. It can also manufacture certain macromolecules, and then it's sort of like a mailroom or like a post office. It sorts and packages materials into these little envelopes called transport vesicles. you can say in summary, the Golgi complex sorts proteins from lipids and package them into these transport vesicles. Now, let's take a look. You see the direction of the red arrows sort of point, so it looks like it's coming at us here, or pointing down or coming at us. And so the arrows that are sort of all pointing down, those are coming from the ER. And do you see how those vesicles are fusing with the Golgi apparatus? And as they fuse, they allow their contents to dump out of them and into the Golgi apparatus. And so those contents, again, kind of like a maze, flow in and out through like a maze, and eventually the contents are sorted and packaged once again into vesicles, transport vesicles on now the trans side, or the front side now, of the Golgi apparatus. And now we're even a little bit closer to the very, very outside, which is the plasma membrane. Again, the Golgi apparatus is made out of the same stuff as transport vesicles, which is made out of the same stuff as the endoplasmic reticulum, which is made out of the same stuff as the plasma membrane. All part of that same system because they're all made of phospholipids. And so they can bind to each other, fuse to each other, and pinch off with ease because they're all made of the same things. Once the packages have left the Golgi apparatus, then the contents can then leave and fuse with the plasma membrane, and if the stuff needs to leave the cell, that can be the end of it. But you also have some other types of packages. A lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. A lysosome is basically one of those little packages, except they contain these enzymes, and enzymes can digest certain proteins or nucleic acids or carbs or lipids as needed. Break it down, and so that we can use the subunits like glucose or amino acids or phosphate and so on. There's ways to take molecules in and ways to take molecules out. If you take large molecules in, the process is called phagocytosis. It's like eating food, and so because the plasma membrane is a very fluid substance made out of phospholipids, it can sort of begin to engulf food, as you see in the bottom left-hand corner there, and wrap around the food molecule, and now the whole thing is all called altogether. It's called a food vacuole, which is a little envelope made out of phospholipids, and inside you got the food. This would be, for example, a protein or a carbohydrate, a long piece of starch. Well, you got to break that down into smaller subunits, which would be the glucose. Glucose is the form that we can use, and so we got to break it down. So the food vacuole then will fuse with a lysosome that contains those hydrolytic enzymes or digestive enzymes, and the two vacuoles then will fuse together, or the food vacuole and the lysosome will fuse together, and the contents will be mixed, the enzymes with the food, break it down, and that's how we can utilize the food coming into each of the cells. Vacuoles are just large vesicles derived from the ER and the Golgi apparatus, all made out of the same stuff, phospholipids. Well, there's food vacuoles, there's contractile vacuoles, And there are central vacuoles, which we generally find in plant cells, to to keep the pressure high inside, and so that way the walls don't cave in, and so that way plants can have structure. Here's a diagram of products being made, starting from the inside of the nucleus, going through the rough ER, smooth ER, on the cis side of the Golgi, and then eventually being transported through the Golgi into the trans side of the Golgi, and then releasing the contents of the vacuole out of the plasma membrane. Mitochondria is the powerhouse of the cell. The mitochondria are the sites of cellular respiration, a metabolic process that uses oxygen, keyword oxygen, to make ATP. ATP is the energy currency of the body, but the mitochondria requires oxygen to even be able to make this ATP. The mitochondria contains an outer membrane, an intermembrane, and in between the outer membrane and the inner membrane is the intermembrane space. The very, very, very center of the mitochondria is called the matrix, the mitochondrial matrix. So you don't just have one membrane, you have actually two, an outer and an inner, and therefore you have actually two spaces. One of them is in between those two membranes, called the intermembrane space. And then, of course, the very, very inside of the mitochondria is called the matrix. We'll get into this in the next unit. That's all you had to know for this unit is that the function of the mitochondria is it's the powerhouse of the cell. It generates ATP for us to be able to go about activities that require energy. The last thing I want you to know is the difference between flagella and cilia. Flagella and cilia. Cilia are little hairs found on a lot of different kinds of epithelial cells, and there's generally a whole bunch of them on one single cell, like 100 on a single cell. 100 cilia on a single cell. What is their function? To move water past the cell, to move mucus, for example, along in the upper respiratory tract, in our trachea. Our trachea is made out of these cells called pseudostratified ciliated columnar epithelium. And the key word is cilia. These contain cilia. And in between those pseudostratified ciliated columnar epithelium, we have goblet cells that secrete mucus. And the mucus is secreted and now lines the trachea. Now all you have to do, what is the function of that mucus then? It traps pathogens in the dust, in the air. It traps it, but now we got to get rid of that mucus, so the cilia actually move the mucus along, and eventually we can spit it out or cough it out, and altogether it's called the mucus escalator. But the point is that those epithelial cells contain cilia, and so cilia are these structures that generally are many in number per cell, and they move things along. On the other side, you got a flagella. There's generally only one or maybe two flagella on a cell, and the flagella, kind of like the cilia, they move, except they move the entire cell. They generally move the entire cell, like the example of sperm. So the single flagella will there's only one or two maybe on a single cell. In the case of sperm, there's only one, and they move the entire cell, whereas cilia don't move cells, but they do move fluids or mucus, for example, past the cell. We did talk about the cell wall. Cell walls only are found on plants, not in animals. Cell walls are only found in plants. And so then remember that, well, what do animal cells have? Animal cells have cellular plasma membranes, not cell walls. And now the last thing that we'll talk about are the 4 cell junctions. 4 cell junctions. We have tight junctions, desmosomes, gap junctions, and Hemi Desmosomes. It's important to note where they are in a typical cell. Epithelial cells have direction. They have an apical surface and a basal surface. And so you notice where the tight junctions are. They're sort of towards the top. They're towards the apical surface. If these cells were made into a tube, for example, a lumen, with those cilia that you see at the top, facing the inside of a tube, like kidney tubules, for example, or in your trachea, then it makes sense that the tight junctions are towards the top, because the first thing that way water that tries to seep through the cells will be stopped by the first type of junction, which is the tight junction. The function of the tight junctions are to keep water or extracellular fluid outside, and to keep water from getting in between those tightly spaced cells, and so that way our skin can stay dry, or that way the skin itself can, for example, can keep water out, and at least for a little bit until our fingers start to prune. But the tight junction, and a good example that I always give is the tight junction is like silicone. Silicone on a window. Silicone is not as strong as screws that you would want to keep that window on, but it does seal well. It keeps it airtight, and it keeps water or rain from getting inside. So the tight junction towards the apical surface, and the tight junctions are like silicone on a window. Desmosomes aren't good at sealing, but they are extremely strong. These ones lock cell to cell. Desmosomes are the strength, and they fasten the different cells together. Those are desmosomes. The next ones are the gap junctions, and these are little pores that allow ions or small molecules to get from one cell to another cell, and so gap junctions are good for keyword communication. Gap junctions allow cells to communicate to each other, allowing molecules or ions through so that one cell can talk to another cell. And the last junction that I'll require you to know is the Hemi Desmosome. It isn't shown here on this diagram, but imagine if a desmosome is a full circle between two cells. It would be like a full circle. Well, it turns out that a hemidesmosome is just half of one desmosome. So instead of being a full circle, it's like a half, and it sits at the bottom of the cell. It basically does the same function, except instead of locking cell to cell, it locks the cell to the basement membrane on the bottom. which would be what all the cells sit on top of, kind of like where the brown area is at the very bottom. The Hemi Desmosome locks the bottom of each of those cells to the basement membrane, and so it has almost the same function as the desmosome. It locks, but it doesn't, instead of locking cell to cell, it locks the cell to the bottom, and that's the function of the Hemi Desmosome. Those 4 junctions I'll require you to know, and remember where, relatively speaking, where they are in the cell, orientation-wise. And that concludes the cellular organization chapter.